Hello, and welcome to the Microbial Insights instructional video and tutorial for using your bioextract kit. Begin by cleaning your work area with an alcohol prep wipe. Now, open your kit. Each individual kit includes the following: 1 alcohol wipe, 2 3 milliliter syringes, a 1 milliliter syringe preloaded with solution A capped, a 1 milliliter syringe preloaded with solution B capped, 1 bead tube, a 1 milliliter syringe fitted with an isolation column, a 1 milliliter disposable transfer pipette, a DNA isolation sample prep cartridge, and a 1.5 milliliter microtube. Also included are 1 pair of needle nose pliers and 1 cardboard microtube holder. After putting on gloves, pull out your first BioFlow filtered sample. Remove the BioFlow filter from the Falcon tube and set the Falcon tube nearby to be used again later. Label the outside of the BioFlow filter to match the sample name on the Falcon tube. Next, label the bead tube found in your bioextract kit to match the same sample name and set it upright nearby. Finally, label the microtube found in your bioextract kit to match the same sample name and place it upright in the cardboard microtube rack. Remove both end caps from the filter and set aside nearby. If needed, use the included pliers to gently loosen the inlet caps. Next, take one of the 3 milliliter syringes and fill it with air. Connect the syringe to the filter inlet and push the air through the filter to expel any water still in the filter. Repeat this as many times as needed in order to remove as much water as possible. Replace the red end cap of the filter. Find and uncap the syringe labeled Solution A in your bioextract kit. Connect the syringe to your filter via the lure lock. Push the solution into the filter by slowly pushing the plunger down to avoid back pressure. Disconnect the syringe. Reconnect the inlet cap and shake vigorously for 5 minutes. If extracting multiple samples at a time, prepare each sample up to the next step that requires shaking and then shake multiple samples at a time. We recommend extracting no more than 6 samples at a time and holding 3 samples in each hand while shaking. After 5 minutes of shaking, remove the inlet cap and repeat the process using the syringe labeled Solution B, found in your bioextract kit. Again, shake for 5 minutes. For the next step, remove the inlet cap from the filter. Pull out the final 3 ml syringe from the bioextract kit and pull 1 ml of air into it. Connect the air-filled syringe to the filter inlet via the luer lock and then invert the filter. Slowly push the air into the filter and then pull the syringe plunger back out to extract all of the liquid from the filter into your syringe. At this point, you are done with the filter, but not the liquid in the syringe. Transfer the liquid from the syringe into bead tubes that you labeled earlier. Close the tube tightly and parafilm the top. Shake the bead tube for 5 minutes. After 5 minutes, set the tube aside, upright, as you continue to the next steps. Next, pull out the sample prep cartridge, followed by the 1 ml syringe that includes the connected column. Use the sharp tip of the column to poke 2 holes into the first section of the sample prep cartridge. Lay the syringe nearby, being careful not to let the tip touch the surface of the table or desk. Pull out the 1 ml plastic pipette and uncap the bead tube. Using the plastic pipette, carefully pull 1 ml of liquid out of the bead tube, avoiding solids as much as possible. Transfer the liquid into the first section of the cartridge by holding the pipette at an angle. This helps to avoid the formation of bubbles and potential spills. Using the 1 ml syringe with the attached column, push the tip into the first section of the sample prep cartridge. As shown on the side of the first section, slowly pull and push the syringe plunger a total of 10 times. Before moving on, make sure the syringe plunger is fully depressed. Do not transfer liquid from one section to the next. Use the column tip to again poke 2 holes into the next section. As shown on the side of the cartridge, push and pull the plunger of the syringe 2 times, being sure to push the liquid fully out before moving on to the next section. Repeat this process with the next 2 sections, colored orange and yellow, each time, pushing and pulling the number of times shown for that section and being sure to push all the liquid out before moving on to the next. For the blue section, you'll be air drying the column. In this section, only one hole is needed. Draw air up into the syringe and quickly pump it out, vigorously repeating this for 20 or more times until the column appears dry. In the final green cartridge section, push and pull the syringe plunger a total of 5 times. Next, pull all of the liquid from this section into the syringe and transfer it into the microtube that you labeled earlier. That's it! You've isolated microbial DNA. If analyzing the DNA immediately, follow the appropriate protocols. If you're shipping the DNA, parafilm the top of the microtube and place it into the correlating falcon tube. Fill out the COC accurately, marking the media as bioextract, and ship it all on ice to the nearest Microbial Insights lab. Upon receipt at the laboratory, Microbial Insights will add the appropriate preservative to ensure that the samples are stable for long-term storage and future analysis.